you. My introduction's briefer than everybody else's. Obviously haven't got as much industry experience yet. Just to say as well, it's a small group, so comments, if you want to interrupt me during the presentation, feel free to go ahead and we can discuss things as we go along, or you can wait to the end, whichever, it's up to you. So my thesis was based at the Drug Safety Research Unit with the in collaboration with the University of Portsmouth. And today I'm going to present on sort of the first phase of that research, and then tomorrow is the second part of it. So this part was looking at MedDRA SMQs and physician ratings of important terms. I looked at the diagnosis of 2 medical concepts, but today I'll just talk about cardiac failure to demonstrate the method. So in the presentation I'll give a brief background to my research, the main research aims. I'm not sure how much everybody knows about MedDRA SMQs, but I'll briefly introduce that and discuss the weighted SMQ that MedDRA currently have. And then discuss the diagnostic process and the therapeutic area and the model drugs that I used to develop my proposed method of signal detection, followed by the Delphi study, which was used for this part of the research, and then the results and conclusions from that. So this is just to say that the project was funded in full by the MHRA, and I was based mainly at this lovely building in Southampton, but also then at the University of Portsmouth, and it was under the supervision of Professor David Brown and the director of the DSRU, Saad Shakir, which I believe some of you know. So yeah, signal detection in PV is important to aid identification of real safety concerns in the large amount of data now available. So it is recognised and widely known that established systems involving the traditional review of individual cases may take too long, and the medicinal product will have reached a large population before signals are detected. So a system is needed where signals are identified sooner so they can be investigated and any causal associations dealt with promptly. This research examined one such system using the yellow card data for modelling and 4 diabetic drugs as experimental compounds. So the main research aims were to review the symptoms, signs and diagnosis that may be associated with conditions considered to be ADRs, and to add a numerical value to those terms according to how discriminatory they are for the condition in question. So the second stage of my research then was to add this numerical value in a form of statistical weighting to the terms prior to disproportionality calculation for signal detection, and allowing a symptom that is strongly associated with the condition to add more value to the qualitative calculations than a symptom that is only weakly associated, and that will be discussed further tomorrow. So when I first started at the DSRU, I came straight from my undergrad degree in pharmacology to the Drug Safety Research Unit and was thrown in at the deep end, and Saad sent me on all of his courses within the first few months and got a load of information, and then after that I had to sit down and be like, well, what am I going to do with all this information? And I think that's relevant to PV in general. Even as far as patients and case reports go, we try to get as much information, not really sure what we're going to do with it when we put it all together. So the background then for MedDRA, as I'm sure most of you know, comprehensive and complex medical terminology developed by ICH. It has been implemented worldwide by drug regulatory authorities and the industry and academia for coding, reporting, analysing and communicating regulatory information, and it is now the most widely used dictionary and has become accepted as an international standard since it was implemented in 1997. So MedDRA SMQs were developed, they're standardised MedDRA queries, and were developed to aid the identification and retrieval of potentially relevant individual case safety reports. So they're groupings of MedDRA terms, usually at the preferred term level, and they relate to a defined medical concept or area of interest, and they can include preferred terms for signs, symptoms, diagnosis, syndromes, physical findings, laboratory or other test data. So each medical concept that is defined by an SMQ is defined by a working group and comes with a list of references and the inclusion-exclusion criteria. and a list of both broad and narrow search terms from various system organ classes. So it can be used on clinical data that are coded with preferred terms from a single MedDRA version. Guidelines have recommended the use of SMQs for signal detection, validation and evaluation. However, there's very little literature on how this can actually be used and the advantage of this for signal detection. So currently there are almost 100 SMQs created, and SAMES recommends that we can use these SMQs at all stages of the PV process. So firstly, the broad search uses the preferred terms in an SMQ and provides high certainty that all cases for a medical concept is being investigated will be identified. And then the narrow search terms used provide high certainty that the cases identified definitely relate to a medical concept. So for some SMQs, there are algorithms introduced to further aid this identification of specific case reports. One example then of a weighted SMQ that has been developed is for lupus. So this came from the fact that there was a great diversity in the terms being reported for 2 drugs that were known to be associated with lupus. And the weight was based on the frequency of reporting retrospectively of these search terms and the probability that they related to lupus. But there's nothing in the literature about the use of this weighted SMQ. So this is just a brief summary of it. So the narrow search terms are 16. So any one of those in a case means that it is a potential case for lupus. And then the proposed weighting. So if the proposed weighting of any of these terms adds up to 6, then it qualifies as a case for lupus. So this was the only literature. Yes? That was developed by MedDRA and their working group. So it was based on the frequency of how often these terms were being reported.
So if it's more than a certain number of ETs?
Yeah, it's being more important. So that's got the higher weightings of 3 there. And then it goes down to 2 and 1.
But I'm surprised. Renal disorder is 8. But it's only weighted as 1.
Renal disorder, that's the number of preferred terms that come under renal disorder. Okay. So all of those 8 terms are giving a weight of 1 each. Okay? So this was the one literature article that I did find about the use of SMQs, and it was a study by EMA researchers that compared the timing of the onset of a known safety signal, hyperglycemia, for a marketed product using MedDRA terms at various levels, and then comparing it with the SMQ for hyperglycemia and new onset diabetes. So proportional reporting ratios over time and dynamic PRR reports for each term level, and then for the SMQ were calculated and used for comparison. And the researchers reported that there was a significant PRR was found using the SMQ earlier than for any event term level studied. So this highlighted the potential for SMQs to be used early on in signal detection for risk management, and that further research in this area would be useful. Now, the diagnostic process. This was a part of my research that I found very difficult, obviously coming from an undergrad, and I had to look at a list of terms and decide how to come up with the weighting and what was more important in terms of diagnosis. So I did quite a lot of research into the definitions of a diagnosis, and there's many definitions out there. So just some of them is that it's a description of a health problem in terms of known diseases, or a set of actions needed to obtain a diagnosis, or a complex transition that begins with the patient's individual illness history, and that the results can be categorized. So just from the dawn of the computer, people have tried to kind of put algorithms along with the diagnostic process. And I know for, like, when I was researching this, there was many sort of examples of algorithms and how med students are taught and how to come up with a diagnosis and lead to things. But the most important part is that even if there's algorithms and all these like computer systems that go along with it, human review is still needed, and that's much the same with causality when it comes to PV. We can try and put all these systems that go along with it to help manage the data, but human review is still what's required to make any decisions. So yeah, that's just a little diagram to show that. So even with all the information, without individual clinical expert opinion, the best evidence may not be applicable or even appropriate to the individual patient. So the agreement of a group of experts through a Delphi study, which is what I come to next, or group discussions is required with higher power than evidence-based medicine. So the Delphi method was first developed in the 1950s. It was originally to forecast the impact of technology on warfare. And the method entails a group of experts who anonymously reply to questionnaires and then receive feedback in the form of statistical representation of the group response, and then the process is repeated, so they're asked to review that group response again. And the goal is to reduce the range of responses and arrive at something closer to expert consensus. And the Delphi method is still used in various fields today. So originally, when I started at the DSU, the idea was that I would go through these list of SMQs and try and work with a physician to assign weighting as to what terms are important and what are not. And that proved impossible, even with all the literature, and it just was taking forever. So then this method was adopted to try and come up with the weightings. So the thought is then that the idea of one physician would be, or the idea was that to get a group of physicians' opinions was more robust than relying on one rater to rate all the symptoms. So, and obviously the physicians with some sort of experience with PV data would provide the best output for this. So to choose the raters, it had to be people that had current or previous history in dealing with adverse drug reactions or some form of PV data. In terms of choosing the medicinal products to model this method, as I was using yellow card data and had access to two separate databases from them, it had to be a product that was on the UK market between 2000 and 2010 and had some sort of new safety signals within that time that I could use to show the method. And also then we had to have a number of yellow card reports with the event terms that we were going to weight. So the risks identified were taken from the scientific discussion from various glitazones on the market between 2000 and 2010. And it was very important to highlight in the letter to the raters to try and ignore what they'd heard in the media since the product had gone on the market. Because for this method, it was the whole idea behind it is that we can come up with weightings based prior to the based on the information available at marketing authorization. So just sort of to summarise, I'm sure most people know the highlights of rosiglitazone. So it was first marketed in the US in 1999. At the same time, marketing authorization was refused in the EU. Then, not even a year later, it was granted in the EU, but with provisions. So it was added to the SMPC that it was contraindicated in congestive heart failure, and they had to have a warning added to the pill regarding the possible development of fluid retention and CHF. And the treating physician had to be experienced in the treatment of type 2 diabetes. And then following this, in 2010, it was withdrawn from the market following EMA review, concluding that all rosiglitazone containing medicines should be suspended from September 2010. At the same time, the FDA decided it should remain on the US market but be more restricted, and a risk evaluation and mitigation strategy was put in place in the form of the RECORD study.
What was the sample size? Any idea about the number of patients who received the medicine and how many of them suffered from heart failure before this medicine was withdrawn?
Oh, sorry, I'm not sure. I've just gone from the main decisions to the end. Okay. So to determine the weighting factors to be applied, the Delphi method was piloted on 2 potential respondents to gain feedback on the layout and just the practicability and just how long the whole process took. Following that, no changes were necessary, and the data from the 2 pilots were included in the final analysis. So then 18 further physicians and 1 cardiac specialist were asked to rate all the terms for the SMQ cardiac failure according to this scale. So 3 if they thought the term was very important in contributing towards the diagnosis of cardiac failure, and so on. And there was also a place for them to put comments on each term or on the method if they so wished. So the results, we had 11 reviewers completed the survey plus the cardiac specialist. Quite a number of terms were left blank, and most of the comments stated that this was due to lack of the specificity of the term. The mean rating for each term was then the mean rating for each term was then worked out along with the standard deviation, and this was then listed, ranked by the mean highest to lowest and standard deviation lowest to highest, and was sent for second review. And only 4 of the 4 original reviewers then responded to this. So that's just a brief summary of the main comments that came back from the raters. So yeah, most of them were saying that agreed that making a diagnosis never relies on a single symptom, sign or investigation, and obviously the bedside manner is what comes into play more so than just rating a term by its importance. So the statistics then used to on this data when we got it all back was to try and assess the proportion of agreement after chance agreement is removed. So we used the Fleece's kappa statistic, and then also the Kendall's coefficient was the strength of the overall agreement within the data set. So for most terms there was little agreement between the 11 raters for all cardiac failure terms, for all 69 terms in the SMQ. And those that were rated as very important had the best agreement, but this was still only fair with a Fleece's kappa value of 0.2. In the second round, there was complete agreement between the 4 respondents, which is expected because they were presented with the ratings that they knew had been gathered from the first round. So I just want to highlight that the respondents obviously were under no obligation to agree with the first round. They were given the chance if they wanted to change or make any comments they could. So it's not quite like a Delphi technique in that if we put everybody in the one room and asked them to agree and everything, you're more likely to agree. So it was completely anonymous and they could write down changes if they wanted. So yeah, we did some further analysis then on the data to compare those clinicians that were currently engaged in PV research and those that weren't currently working with it. So the agreement for those who work in PV research was better, as in they were all slight, but it still wasn't great. And then the agreement of the 11 raters compared to the single consultant cardiologist improved greatly. So the cardiologist agreed with the overall means for those terms rated 3 with showing moderate agreement. So in conclusion, it should be noted that the levels of the rating scale were relative to each other but not absolute. So they were ordinal in nature and limited the level of statistical analysis that could be performed and hindered comparisons with other rating scales and exercises. But it was aimed to obtain expert opinion on what was considered important signs and symptoms that could be used to facilitate signal enhancement. So the process could combine both clinical and scientific judgment. Within pharmaceutical companies, all the information about a medicinal product from the initial discovery phase right through to the application for marketing is available. So that would perhaps make this method a bit easier, and that I was only relying on the scientific discussion that I could get from the EMA website at the time of marketing. And then this information would be ideal for the proposed method. So tomorrow, sorry, I've kind of skipped through that quite quickly, but tomorrow then I describe a bit more how I use these weightings for potentially enhancing signal detection methodology. So is there any questions or comments? Yeah.
Is it common with the Delphi technique that you have such a dropout from the first to the second round?
I'm not sure, to be honest. I mean, it wasn't ideal at the time, but I think given just the time settings that I had, it was quite rushed in that so I'm not sure, to be honest, what the dropout rate would usually be. But I mean, I sent the list with all the means and standard deviations to everybody that replied in the first round, and that's just the 4 replies we got. So maybe it was a case that they agreed with all the means or just felt that there was no need to comment further. I'm not sure. So.
You mentioned that MedDRA have done sort of some work on a weighted SMQ.
Yeah.
Again, a question that came from behind in
Yeah.
terms of the renal component, which is fairly high in lupus and
Yeah.
generally tends to be where it
Yeah.
tends to manifest. But interestingly enough, do you
We'll
have
go
any
back
idea
to
of
it.
what their plans are in MedDRA to progress any further? Because
Yeah.
I mean, from a review perspective, you stuck between a rock and a hard place because you use a broad search and you get too much.
Yeah.
And you use a narrow search and you miss stuff.
Yeah.
So
Yeah. Like, I think the concept behind a weighted SMQ is great, but it's just getting the weighting right.
Yeah.
And so that you don't miss too many or identify too much noise.
Because what you
So-
at the moment is you using a broad search because you have to, because you may miss
Yeah. Yeah.
1 or 2 cases of them.
Yeah.
We all know 1 or 2 cases can make a break.
Yeah, exactly, yeah.
So you're doing a broad search, and it still does then boil down to the fact that you actually need to look at each individual
Yeah.
case, which is it's almost defeating the purpose of aggregate signal
Yeah.
detection. And
Yeah,
monitoring.
I agree
It
completely.
is very, very very labour intensive.
Yeah.
I was just wondering if there are any further plans, but it's good to know
Yeah.
that it's been considered.
Yeah. So, like, when I first started the research, I was aware of MedDRA, but I wasn't really aware of their SMQs and how they'd been used. And then it was actually at a conference through the DSRU that someone came and spoke to me that works and does the training for MedDRA, and she was like, do you know, we actually have, because I was just talking about weighting terms and weighting important terms, and she was like, we actually have algorithms in place for some SMQs. There's one weighted SMQ. We're talking about maybe developing more. So I know it's something that they've considered, but obviously there hasn't been much research into how it can be applied and used. So yeah, they were very interested then to see if it's applied to more data, and obviously if they have those results, then maybe they'll progress and have more.
Okay, thank you very much, Michelle.
Yeah.
One question regarding
Yeah, sure.
In the part 2 of your research, where there's 100% agreement between the 4 raters,
Yeah.
it seems like that there seems to be bias. How did you control for bias between the part 1 and the part 2? Because I think from part 1 you kind of already skimmed the top.
Yeah.
And does the part 2 rater know that it's already been?
Yeah. There were the 4 people that replied to the second one had done the first round.
Okay.
And
But are they aware of the results? that you're
yeah,
presenting?
yeah, they were provided with all of them, all the results for all 11 raters, and then the mean. So
I
yeah.
was just wondering if the second part was biased because they already know.
Well, it was quite biased then that I had 2 of the second round reporters who did reply and do the second review ask me which, because I just had it by number, raters 1 to 11, and they asked which rate was theirs to compare it to the mean. And then they decided not to make any changes. So I guess it is biased in that way that they were like, yeah, okay, it goes with that. But I think it's quite difficult to do that in a Delphi study, but yeah, so.
Good, thank you very much.
Thank you.